Well, thank you much. Before I jump into things, Jeff, I hope you don't mind if I take a minute just to make a comment. I've been here for 24 hours now as a member of the External Advisory Board and spent the greater part of the day in meetings with the IDRP. And I just have to say that it really is impressive. I mean, I can't tell you how impressive this is. This is the way it's supposed to happen. You have a first-rate institution with a supportive dedicated community, bring in a world-renowned investigator, and you create something out of nothing. But you folks, and I mean this, Jeff, as a compliment to you and to the institution, and I also mean it to you folks as well, since you've largely made this possible. You haven't just created something out of nothing. You've created something incredible. So I really need to salute all of you. Now, I know a lot of you want to hear about what's coming up in the pipeline in terms of Alzheimer's treatment. And Dr. Schmidt is going to discuss some of those, is going to discuss the pipeline immediately after me. But what I'd like to do is just take a few minutes and set the stage for what he's going to tell you. It will help put Alzheimer's disease clinical trials into some perspective. So the first slide just talks about the generic phases of drug development. So phase 1 studies of drugs are when you are simply testing a new drug that you think might be helpful in a disease. You're testing it in young, healthy people just to see whether it's safe. And if young, healthy people can tolerate the drug with not too much going wrong with them, the drug will often then go into phase 2 trials. Phase 2 trials are also safety trials primarily, except they're safety trials in populations of who you want to treat. So you might test your Alzheimer's drug first in young, healthy people in phase 1 if it's tolerated. Then you might test it for safety in people with Alzheimer's disease. Now, although phase 2 trials are meant to be primarily safety trials, everyone kind of wants that little sneak preview about, is the drug maybe really going to work? And so there's a sort of a trial called the phase 2B trial, which is really intended to look at safety, but measures of efficacy will be evaluated during the trial, and at the end of the trial, the company that's hoping to develop the drug will hopefully have some kind of idea about how likely it is that their drug is going to work. And if the signal looks good, if things look promising, then they'll move forward into a phase 3 trial in which they're testing now the drug in the Alzheimer's patients, but the primary focus of the trial is to determine definitively whether or not the drug works. And if it does appear that the drug works based on how the company defines success, and they define what's going to be success before the trial's done. If they meet those criteria, then hopefully the drug will be approved by the Food and Drug Administration if they feel that it's safe and efficacious. Phase 4 studies are studies that investigators may do on a drug after the drug has been approved for use and is out there. For example, Alzheimer's was originally approved for the treatment of mild to moderate Alzheimer's disease from successful phase 3 trials. And then later on, people went and did phase 4 trials on it to see whether it had any efficacy in severe Alzheimer's disease. Now, how do you judge when you're doing a clinical trial whether there's an effect of the drug? Well, the first point I need to make is that Alzheimer's disease is a chronic and progressive disorder. Over time, people's memories and their cognition tends to get worse. So if this is time on this axis, and this is mental abilities on this axis, and this is going in this direction, is getting worse, over time, we expect people to decline on tests of memory and in memory function and in daily living function. Ideally, we would like to have drugs that really get in there and affect what's primarily wrong in the brain, that are causing the neurons to have dysfunction and to die. And if we find a drug that does that, well, that would be called a disease-modifying effect. And it would be characterized in a trial, and what we look for in a trial when we're looking for a disease-modifying effect is that the trajectory of the decline is changed. So people are getting worse at a slower pace. So this suggests that we probably have affected some fundamental disease process. If we affect it a lot, maybe what we would get is a flat line here. That would sort of be a cure. Sometimes what we've seen in past trials, and certainly what we've seen with drugs like Aricept and Razodine, Exelon and Namenda, is what we call a symptomatic effect. And that is people get put on the drug and they get a little bit better. So this is getting worse and this is getting better. Okay? So something's happening to sort of make these people, that's really benefiting the patient's symptoms. It may be affecting the underlying disease process, or it may just be doing something that temporarily brings a benefit. If we continue to follow those people in the clinical trials, and we see that, yeah, they get better initially, so that's the symptomatic benefit, and then they start to decline again, and this line is parallel to this line, then we interpret this as meaning that there really is only a symptomatic effect. So, and this is really what happens with Aricept. People get on the drug, they may look a little bit better, then they start to decline again. About a year after they start treatment, they're coming down below their baseline. It doesn't mean that the medicine isn't working anymore, but the people are declining below their baseline at a rate that you'd expect them to decline, only they're doing better than they would if they weren't taking the medicine. This would really be great if we were able to accomplish this. A drug that had both symptomatic and modifying effects, so getting better again is benefiting the symptoms, and then having a slower decline is affecting the actual physiology of the disease. Now, there are a number of problems that we need to be aware of when we design Alzheimer's clinical trials. So we realize that the effects that our drugs may have may be subtle. So the way you get around, the way you gain the ability to see a subtle effect is to follow enough people for a long amount of time. The more subtle the effect, the more people you need to follow and the longer you need to follow them for. Also, people tend to drop out of trials, especially if the drug has unwanted side effects. And it's not entirely clear what the best way is to include or exclude data from people who drop out of trials. It can actually have quite an influence on how the trial is ultimately interpreted. Okay. Placebo and practice effects. Practice effects means on the memory testing. So some people, if they take memory tests enough, they actually start to get better on the tests. And so it can make it look like that the drug's having a benefit, but it's really not. It's a practice effect. So we have to be careful when we design these trials that we take into account the possibility of practice effects, and also that you can put someone on a placebo and they'll initially look better as well. Subjects, especially in 2009, may be taking other medications that can impact results. So now everyone wants to be on the drugs that have been approved by the Food and Drug Administration. Can you put someone in a clinical trial for a drug that hasn't been proven yet, with the hope of proving it, and they're already on an Alzheimer's treatment? Well, ethically speaking, that's really where the field has gone, but we always wonder if the medications that people are taking while they're in the trial may be actually impacting the trial. Also, the subjects in the trials can vary quite a bit among themselves, so we have to be careful about variations between the groups. And then also, these are multi-center trials usually, and the quality of the data may vary between centers. So you may have a center that's very good at doing the trial, and they generate very good data, and another center may be, garbage in, garbage out kind of place. So there are lots of things that can confound a trial, so ideally we'd like to think that they go smoothly, but often they don't. Before something makes it to a clinical trial, though, it has to go through preclinical development. So how do we come up with things for preclinical development? Well, what we do is we create a paradigm. A paradigm means what is basically asking the question, what's causing the disease? And then if we can come up with what we think is causing the disease, then we can try to come up with interventions that we will test in models of the disease. And these can either be things that we look at in cultures of cells that we grow in flasks, or we can design animals, transgenic animals that sort of mimic aspects of the disease that we want to treat, like Alzheimer's disease. And then after we've made a mouse that we think has the disease that we want to treat, we can go and try and treat that mouse. And if something works, we can then, it supports moving it forward in human trials. The paradigm in Alzheimer's disease for the last 15 years or so has basically been that there is this protein called beta amyloid that is cleaved out of a bigger protein, and it begins to clump up, and as it begins to clump up, it kills brain cells. And so scientists have made mice that have problems in which they produce this beta amyloid protein, and these mice get plaques in their brains, and when they get older, their memory isn't as good as when they were younger. And so we test treatments in mice like this to try and make the mice better. The only question is, this is great if we're barking up the right tree. If we're barking up the wrong tree, then we're wasting a lot of time finding that out. Because it may turn out, if this mouse isn't accurately modeling Alzheimer's disease, we may come up with something to cure this mouse, which will be great for this mouse, but it might not work in people. And unfortunately, there's a lot of words on this slide and on this slide. I just want to make the point, there are now unfortunately, there are a number of things that have worked in studies of these mice that have now been tested in humans and have not worked. Arguably, the one that I suspect the one you've probably heard the most about is this beta amyloid vaccination study that was started a number of years ago, and it was stopped sort of in the middle of the study because some people were developing an inflammation of the brain. But as the people who were in this study were followed for years at a time, it turns out that we realized that those who got this vaccine actually had the beta amyloid removed from their brains, which really was great. It's amazing that we can accomplish this. The only problem is the people appeared to decline at about the rate that you would have predicted they would have declined if you hadn't gotten the beta amyloid out of their brains. So, all this helps us learn, and we'll get better at this. But the bottom line is it may turn out that there are at least two kinds of Alzheimer's disease. An early onset familial form where people tend to get it in their 40s. Dr. Sheff was talking about this earlier. And the mice that we study Alzheimer's disease in are really modeling this type of Alzheimer's disease. And the treatment that we're developing is really intended to try and treat whatever disease this is. What most people who walk into my clinic have is late onset sporadic Alzheimer's disease, about 99.99% of the cases. And really one of the questions that we need to all agree upon, and we're not in the field isn't in complete agreement, is this disease and this disease, do they have different bases? So with that, I will finish and turn it over to Fred, I think.